Nano-drug delivery.
Medicine via nanobots.
Also, I'm David Masterling.
I'm Kareem Harris.
Alright, so what are we doing with this project? Pretty much we're trying to find an alternative to cancer treatment. Now, traditional cancer treatment has shown results. It's worked, but with, for example, chemo, it affects everything. Any cell that kind of splits in half. So we're trying to find something that is more specific.
And the significance of this is the fact that if we're able to actually do this, we might be able to make a broad treatment that can go towards not just things such as cancer treatments, but any kind of treatment, any kind of disease that has to do with the DNA.
All right. So what is the purpose? What is the purpose? What is the dream? It's just nanobots, small things we can inject into someone that would be able to target in this instance, cancer, and treat it.
And our hypothesis, it just follows that it's if we can apply this concept of nanobots to delivering DNA or drugs at the nanoscale, then we can cure diseases at the cellular level.
All right, so we looked into different methods of doing this. The first one was actual, little nanobots, things almost like little machines. Now they showed some promise. However, the main problem was biocompatibility. You can't really put these little robots in someone. The immune system may freak out. You're putting a lot of metal in the body, which could actually kill the host. So
that's something to look into. I mean, we're putting it as a research path. So we definitely want to look into the possibilities of that, but we've gone towards going a more biological route, which we'll have on our next slide. But we do want to look at photosensitive linkers, which are very much around nanotechnology, because that has to do with shooting light to activate nanoparticles, which that's a whole thing in itself. Blood biology, again, material blood. We don't want to kill a patient.
Also, what we're working on is the microscale, but all the components we're using are on the nanoscale. So pretty much you have a bunch of little tiny parts, and they all end up being around one micrometer.
Yep, and that's also the size of the bacterium, which is 0.1 to 10 micrometers, which we have the numbers here. 1 times 10 to the negative 9 meters is a nanometer, and to the negative 60 is a micrometer.
All right, so this is what showed the most promise. DNA origami. As the name kind of suggests, it's the art of folding DNA into some sort of shape to serve a purpose. Now, for us, we're looking for kind of like a barrel shape. And what this allows us to do is kind of encapsulate our medicine to be released.
And we have an example here. We'll get back to that in a second. And our spin on this is something called the jar, which encapsulates that DNA origami to protect it further, while the origami, as you can see here, is in a hexagon, which we found is nature's perfect shape. We want to protect that DNA further. Sorry, we'll get to that. And this would be powered by ATP, which, look at that, that's something that the body already uses as energy. And we're thinking about a lipid bilayer, just a lipid layer really, that's nanometers in width, which again, that's going more the biological route. And chaperones, which are proteins, part of a group called molecular chaperones, which help DNA unfolding. So it's kind of the perfect pair. And then this is the example where, really quick, we have DNA that it's folded. Then this is a shape, and there are these things called aptamer locks, which are unlocked by the antigens found on certain cells. When that DNA origami is unlocked, it connects to whatever cell it's responded to via antigen, and it connects, and the payload allows cellular signaling to change whatever the cell's doing. So these are the companies we looked into. One of them is called Leidos, which is a joint share of Lockheed Martin, which Poly's partnered with. And they're very much less military, more so in the medical field, particularly precision medicine, which, nanometers, micrometers, very precise if you ask me.
Also, we have nanocomposites who actually supply for Polytechnic and a lot of our supplies, so definitely could help us out.
Which goes to an experiment we did in our nanoscale class last year. So we do have a connection to this company. Government agencies include those such as NASA, the National Institute of Health, the National Science Foundation, because there's actually a thing called the National Nanotechnology Initiative where the government actually apportioned certain parts of these agencies' budgets to go towards nanotech research and development. We also have other companies that are more so just for providing and aiding in promotion and research, but mostly we want to look into those. And as far as anything like what we would have by the end of, by the expo would probably be a diagram, maybe a 3D print, possibly.
Good. All right. Questions, guys?
1 minute.
Okay. Questions? All right. So how are you gonna do the design? If you are gonna go into the 3D printing of this, right?
Yep.
How are you gonna make the design of the layers? Are you gonna have one layer printed out as the membrane, and then because then it's not gonna be visible, right? How
Right.
exactly is the
So we're thinking maybe something where we can cut it open, or maybe just print half, or maybe even an exploded view where it's just different parts. So then if you put it together, you have
right?
one thing.
That might be the
Yeah.
best. I would go for that.
Yes.
Do it.
What are your names again?
Kareem Harris,
And David Maslin.
What group number are you?
number 8.
Okay. Thanks.
Question there, Nick?
No, I was one script number.
Okay.
Also, the citations didn't show up in the PowerPoint. So
Yeah.
Yeah, I know. I was like, where are your citations? Because again, remember that's you gotta show that.
These are all of our citations right here.
Okay. Okay.
Quite a bit.
Yeah, they did a lot of research on this.
Yellow
So,
field, lots of information.
any other questions? None? All right. Thank you, Chris.